picture this. You're standing on the edge of a vast sweeping field of sugarcane.
Okay.
The wind blows, the stalks sway, and as you look out across the acreage, every single plant looks, virtually identical.
Right, just a sea of exact replicas.
Exactly. The height, the leaves, the color. But now take that image and contrast it with, say, walking into a crowded coffee shop or just looking around a room full of your friends and family.
Oh, yeah. It's a completely different experience.
Right. You see this unbelievable explosion of variations, different heights, different eye colors, different hair textures, face shapes.
It is a really profound visual contrast when you put them side by side like that because, it forces you to confront a biological reality that we usually just ignore.
Yeah.
We completely take for granted that human beings look distinct while we just, expect a field of crops to be uniform.
And understanding the mechanism behind that contrast is basically the core mission of today's deep dive. We've been cross-referencing a stack of evolutionary biology journals, some historical texts on the origins of genetic math, and cutting-edge research on chromosomal inheritance.
It's a lot of material to cover.
It is, but we're doing it to answer a fundamental question for you. How exactly are all these variations created? Like, what are the microscopic invisible rules that dictate why you look a bit like your parents, but not exactly like them?
Right. It's the ultimate puzzle of heredity. We're essentially looking at the biological architecture that somehow balances the need to keep a species functionally consistent with the absolute necessity of making each individual unique.
Okay, let's unpack this. Because to understand how traits are handed down through generations, we first need to figure out how a variation physically comes into existence in the first place.
Exactly.
So we have to start with the baseline of reproduction. In that sugarcane field, or say in a colony of bacteria, you're looking at asexual reproduction.
Yeah, that is the baseline. So imagine a single bacterium. When it reproduces, it simply replicates its internal material and divides.
Just splits down the middle.
Right. One bacterium splits into two, those two into four, and so on. Because there is only one genetic parent involved, the offspring are inheriting a singular, identical body design. So, for all practical purposes, they're clones.
But the research points out they aren't flawless clones. Even in asexual reproduction, variations happen. They're just, incredibly subtle.
Very subtle, yes.
I was trying to conceptualize this, and it feels a bit like taking a beautifully typed document and running it through a photocopier.
Okay, I like that.
And then you take that copy and you run it through the photocopier again. If you do this hundreds of times, the text fundamentally stays the same, but maybe tiny specks of dust get on the glass.
Yeah.
Or the ink smudges just a fraction of a millimeter, and those tiny inaccuracies just accumulate.
That's a great analogy. The biological equivalent of those specks of dust are essentially errors in DNA replication. When a bacterium copies its DNA, the polymerase enzymes doing the molecular copying are incredibly accurate, but they aren't perfect. They occasionally insert the wrong chemical base pair.
So it's a typo.
Exactly.
Yeah.
So the resulting bacteria are nearly identical, but they carry those microscopic chemical smudges.
Wait, I have to question the underlying logic of that system, though.
Yeah.
Because if the creation of new variations relies on random copying errors, isn't that like an incredibly dangerous survival strategy?
Oh, absolutely.
Because if you randomly change a line of code in a complex computer program, you almost never improve it. You usually just crash the software.
You've identified the inherent risk of mutation right there. The vast majority of those random copying errors are either entirely neutral, meaning they do absolutely nothing, or they are actively harmful.
Right, they break the system.
Yeah, a mutation that breaks a vital cellular function will simply kill the organism. But you have to remember, the environment acts as a relentless, unfeeling filter. It doesn't care about the broken variants. It only cares about the rare error that happens to confer a structural advantage.
So the environment is basically stress testing every single copy error.
Exactly. Let's look at the specific heatwave example from the evolutionary biology journals we reviewed. Let's say one of those random DNA inaccuracies alters the lipid composition of a bacterium's cellular membrane.
Okay.
Under normal conditions, this altered membrane might actually be a liability. It might require more metabolic energy to maintain, which makes that specific bacterium slightly less efficient than its peers.
So it's carrying a penalty when times are good.
Exactly that. But then a severe heat wave strikes. The normal bacteria, the ones with the standard, highly efficient membranes, they can't withstand the thermal stress.
Oh, wow.
Yeah, their membranes literally melt and rupture. They die off rapidly. But that one variant, the one carrying the quote-unquote inefficient mutation, its altered lipid structure, happens to have a higher melting point.
So it survives the heat wave.
It survives, and because the competition is suddenly gone, it thrives and multiplies, passing that heat-resistant membrane to millions of offspring.
That's wild. So the variation didn't happen as a response to the heat wave.
No, not at all.
The mutation was already there, just hovering in the background as a random error, and the changing environment suddenly made it the most valuable trait in the room.
That environmental filtering is the mechanical engine of evolution. The environment dictates which random errors are suddenly advantageous.
Okay, so that explains the why of variation. It's essentially a species randomized insurance policy against a volatile world.
Well put, yeah.
But that only explains how a single, simple trait survives in a clone. It doesn't explain the rules of how complex traits are handed down in organisms that use sexual reproduction.
Right, which is a whole different ballgame.
And for that, we have to look at the historical texts on Gregor Johann Mendel.
Mendel is such a fascinating historical anomaly. I mean, he was working in a monastery in the mid-19th century, decades before the scientific community had any concept of DNA or chromosomes or molecular biology.
It's a great irony that the foundational math of modern genetics came from someone entirely outside the scientific establishment.
Yeah, it really is.
Like, the historical records show he actually failed his teaching certificate exams. He wasn't a recognized academic superstar. He was just a monk in Brno who started rigorously crossbreeding garden peas.
And honestly, his outsider status might have been his greatest asset. Because the prevailing biological theory of the 19th century was blending inheritance.
Which means what exactly?
It's the idea that traits from two parents mix together like liquids. So if you cross a tall plant with a short plant, the scientific consensus was that you would get a medium height plant.
Which kind of makes intuitive sense, right? Like mixing paint.
Right, but Mendel didn't approach this as a traditional biologist. He approached it as a mathematician. He utilized discrete mathematics and probability to track traits across generations.
Okay, let's skip the high school biology textbook version of the Punnett squares and get to the core of what his math actually proved.
Sure. Yeah.
Because he crossed purebred tall pea plants with purebred short pea plants.
Yeah.
And in that first generation of offspring, there were absolutely zero medium height plants.
Right.
Every single one was tall. The short trait seemingly vanished.
Which completely shattered the blending theory right there. But the true mathematical breakthrough came when he let those tall offspring self-pollinate.
And what happened?
In the next generation, the short trait reappeared in exactly 1/4 of the plants. That specific 3-to-1 ratio proved that the shortness trait hadn't been diluted or destroyed. It had been carried invisibly.
This completely reframes how we think about inheritance. It's less like mixing paint and more like having two light switches wired to the exact same bulb.
Oh, I like that.
Yeah, so if the tall trait is the dominant switch and the short trait is the recessive switch, it doesn't matter if the short switch is flipped off. As long as the tall switch is functioning, the light turns on. The plant grows tall. The only way the room stays dark, meaning the only way the plant stays short, is if both switches are broken.
That light switch analogy translates perfectly to the underlying chemistry, even though, Mendel couldn't see the chemistry himself.
Right, he had no microscopes powerful enough for that.
Exactly. But he deduced that there must be discrete physical factors controlling these traits, and that every organism carries 2 copies of these factors for any given trait, one inherited from each parent. We now call those factors alleles, or variants of a gene.
So what does this all mean? How do we go from a conceptual mathematical factor to a physical leaf or stem? Because Mendel's math proves the traits don't blend, but math cannot physically construct a plant.
To understand the physical construction, we have to bridge Mendel's math with modern molecular biology. We have to look at how DNA acts as an information architecture.
Okay.
Cellular DNA is a massive sequence of chemical code, and specific sections of that code genes contain the precise instructions for assembling proteins.
So if DNA is the recipe book, then the gene is the recipe for a single dish.
Perfect.
But how does that dish determine if I have free or attached earlobes, or if a pea plant is tall or short?
Let's stay with the plant height example. The physical height of the plant is dictated by the amount of a specific growth hormone it produces.
Makes sense.
But that hormone doesn't just appear out of nowhere. It has to be synthesized by an enzyme. And an enzyme is simply a specialized protein functioning as a biological machine.
Okay, let's build on the recipe analogy then. The DNA is the recipe book. The enzyme is the pastry chef reading the recipe. And the growth hormone is the cake they are baking.
Okay, tracking with you.
If the recipe is perfectly clear, the chef works quickly, bakes a massive amount of cake, and the plant have plenty of hormone to grow tall.
Yes, and let's zoom in on what happens when there is a typo in that recipe, which represents a mutated recessive allele.
Oh.
Right. That typo doesn't just confuse the chef, it physically alters the shape of the chef's hands.
Wait, really?
Yeah, because proteins function based on their three-dimensional shape. If the genetic code has a mutation, the resulting enzyme folds incorrectly. And because it's misfolded, it literally cannot bind to the precursor molecules it needs to synthesize the growth hormone.
So a dominant trait isn't overpowering a recessive trait like a bully.
Not at all.
It's just that the dominant gene is producing a functional pastry chef, while the recessive gene is producing a chef who can't bake.
Exactly.
As long as you have at least one working chef, one dominant allele, you get enough cake to grow tall. You only end up short if both your copies of the recipe are defective.
That is the physical mechanism behind Mendel's discrete factors. And the complexity multiplies when you realize that every sexually reproducing organism has thousands of these traits, governed by massive lengths of DNA organized into distinct structures called chromosomes.
Which brings up a critical structural issue, because if I have 2 copies of every chromosome, one from my mother, one from my father, how do I pass my genetics on to a child without them ending up with 4 copies?
Right, it would just keep doubling.
Yeah, and then their child ending up with 8. It'd be chaos.
The body solves this through a specialized cellular division that creates germ cells, so sperm-egg cells. During this process, the total number of chromosomes is halved.
Oh, I see.
The germ cell only takes 1 chromosome from each inherited pair. Then, when the sperm and egg later fuse during fertilization, the resulting embryo has the correct stabilized number of 2 copies per pair.
Okay, but I need to push back on the logic of how that division happens, because the research emphasizes independent assortment, meaning the germ cell randomly grabs either the maternal or paternal chromosome for every pair.
Yes, it's completely independent for each pair.
But randomly grabbing chromosomes seems incredibly reckless. If the sorting process is completely blind, isn't there a massive risk of a species losing vital traits in a single generation?
How do you mean?
Like, what if a sperm cell randomly fails to grab the chromosome that contains the instructions for building a heart?
Oh, I see where you're going. Yes, that would be fatal. But biological architecture prevents it through the use of homologous pairs. You don't have a random jumble of chromosomes. You have distinct numbered pairs.
Okay.
You have 2 copies of chromosome 1, 2 copies of chromosome 2, and so on. Both copies of chromosome 1 carry the genes for the same types of traits. Let's say eye structure or heart development.
Oh, I see. So it's not randomly reaching into a giant grab bag of all possible genetic instructions.
Yeah.
It's reaching into pair number 1 and taking a copy, then reaching into pair number 2 and taking a copy.
Precisely. The germ cell is guaranteed to receive a complete manual for building a human being. The randomness only applies to which edition of the manual it gets for each chapter, the maternal edition or the paternal edition.
Wow.
And that blind, independent sorting is what ensures incredible genetic diversity, creating trillions of potential combinations while guaranteeing that no essential structural blueprints are ever left behind.
Here's where it gets really interesting. Because we've established this perfectly balanced, highly regulated system for sorting inherited traits. But how does this exact same mechanism determine something as entirely fundamental as whether an individual develops as male or female?
Sex determination is one of the most fascinating areas of evolutionary biology, largely because nature has not settled on a single universal mechanism.
Really?
Yeah, different species have engineered wildly different strategies to answer that exact question.
The examples in the research regarding reptiles are stunning. Like, in several species of turtles and crocodiles, sex determination completely bypasses genetics.
Completely.
It is entirely dependent on the environment. The temperature of the sand where the eggs are buried literally decides if the hatchlings will be male or female. How does ambient temperature override genetics?
It all comes down to a temperature-sensitive enzyme called aromatase. Aromatase is the biological machine responsible for converting male hormones, like testosterone, into female hormones, like estrogen. In these specific reptiles, the gene that produces aromatase is thermally reactive. At cooler temperatures, the gene remains largely inactive. Very little aromatase is produced, the testosterone remains unconverted, and the embryo develops as a male.
But if the nest is hot,
If the environmental temperature rises, it triggers the gene to ramp up production. Aromatase floods the developing embryo's system, aggressively converting the testosterone into estrogen and driving the developmental pathway toward female. The environment is literally throwing the biological switch.
that is just incredible. And the sources also mention marine snails, like the slipper limpet, which undergo sequential hermaphroditism.
Yes, the snails are a wild example.
They're born male, but if they settle on top of another male, they physically alter their biology and change into a female. It proves that in vast sections of the animal kingdom, biological sex is fluid and environmentally responsive.
It really is.
But humans are entirely different.
Yes.
For us, it's not about ambient temperature or social environment. The blueprint is locked in the chromosomes.
Yes, human sex determination is rigidly genetic. Earlier, we established that humans have pairs of homologous chromosomes. To be exact, we have 23 pairs.
Right.
The first 22 pairs dictate the vast majority of our bodily structures. But that final 23rd pair contains the sex chromosomes, and they are the lone exception to the rule of perfect matching pairs.
Right. Women possess a matching pair of sex chromosomes, which scientists label X and X, so women are XX.
Mm-hmm.
But men have a mismatched pair.
Okay.
They have one normal-size X chromosome and one incredibly short one labeled Y. So men are XY. I was looking at the genetic sequencing data for that Y chromosome in the source material.
It's surprising, isn't it?
It really is. Compared to the massive X chromosome, the Y is essentially a shrunken, degrading piece of genetic code. It has lost thousands of active genes over millions of years of evolution. It is a brilliant bit of scientific irony that this degraded fragment of DNA acts as the ultimate coin flip for male development.
It is diminutive, absolutely, but it packs a highly specific targeted punch. Because a mother is XX, every single egg cell she produces will carry an X chromosome.
So she can only give an X.
Right. The female genetic contribution is a biological constant. The father, however, is XY. When his body undergoes that cellular division to create sperm, half the sperm receive the X and half receive the Y.
So the father's biology is essentially flipping a coin millions of times over.
Exactly.
If an X-carrying sperm fertilizes the egg, the resulting embryo is XX, developing into a female. If a Y-carrying sperm fertilizes the egg, the embryo is XY, developing into a male. But how does that tiny, degraded Y chromosome actually force the body to build male characteristics?
The power of the Y chromosome lies primarily in a single critical master switch. It's called the SRY gene, the sex-determining region Y.
Okay.
The SRY gene produces a specialized protein that functions as a transcription factor. This means its entire job is to scan the rest of the DNA and turn other genes on or off.
Oh, I see. So it's an executive manager. It doesn't do the physical building itself. It just activates the biological assembly line.
Exactly. Without the SRY gene, the default developmental pathway for a human embryo is female. Ovaries develop, and the hormonal cascade supports female physiology.
Wow.
But if that tiny Y chromosome is present, the SRY protein binds to specific regions of the genome. It aggressively turns on the genetic pathways for male development, initiating the formation of testes, and simultaneously suppresses the genetic pathways for female development.
So the entire physical divergence between male and female biology rests on the presence of that one microscopic executive manager protein overriding the default setting.
Yep.
It is biological math operating with just ruthless precision.
And because the father produces those X and Y germ cells in a perfect 1-to-1 ratio, the statistical outcome across the human population remains perfectly balanced, half male, half female, driven entirely by the mechanics of independent assortment that Mendel theorized over a century ago.
It fundamentally changes how you look at the world. Let's trace the arc of what we've uncovered today. We started by looking at a field of identical sugarcane, realizing that asexual cloning leaves organisms entirely dependent on microscopic copying errors to survive environmental stress.
Right.
We discovered a 19th century monk who used discrete mathematics to prove that our varied traits aren't blended like paint, but inherited as distinct physical factors. We zoomed into the molecular level to see how mutations physically alter the folding of enzymes, determining whether a plant grows tall or a human has free or attached earlobes. And finally, we explored how a single master switch gene on a degraded Y chromosome overrides a biological default to determine our sex. Every variation you see in a crowded room is the result of this ancient molecular math.
It is an elegant and durable architecture for sure, but it is deeply reliant on time, which leaves us with a critical biological question to consider. Well, we know that environmental pressures like a sudden heat wave act as the filter that selects which inherited variations survive. The environment tests the mutations. But what happens to a complex species when its global environment begins to change at a rate drastically faster than its generational reproductive cycle can naturally generate new genetic variations?
When the climate shifts faster than the evolutionary copying errors can keep up, that is a sobering and vital question to ponder. Well, thank you for joining us on this deep dive. The next time you find yourself looking at a uniform field of crops or scanning the incredibly diverse faces of a crowd, take a moment to appreciate the invisible, relentless biological math operating right beneath the surface. Keep questioning the incredible mechanics of the world around you.